Hello, welcome everyone. My name is Arvind. I'm faculty member in bioinformatics, and it's my pleasure to welcome and introduce Dr. Ruben Dries from Boston University Medical Center. Dr. Dries is an assistant professor in the Department of Medicine at Boston University and affiliated with the sections of hematology and oncology, and also computational biomedicine. His lab and research are focused on implementing the latest spatial technologies and methods to create and analyze multimodal dataset that can bring a new lens to tumor initiation and understanding tumor progression. His work is performed in close collaboration with clinical collaborators at the Boston Medical Center and aims to reduce or eliminate cancer health disparities for our diverse patient populations. Ruben is also the inventor of the Giotto Suite. You will hear about it more. It's a fantastic software, lots of interesting capabilities. I'm very, very eager to hear about Ruben's and his group's work. Welcome, Ruben.
Thank you so much. Can everybody hear me? Is it okay? No? Yes? Should I just speak up? Would that work? All right. Let me try to do just that. All right. So I'm going to talk to you about Giotto Suite today. First of all, thank you for inviting me. It's been really great so far, and I love the kind of like the atmosphere and the space. So first, a little bit about what my lab is doing right now. All right. So my lab really is centered about spatial biology, and we try to tackle it from different points of view. So today I'm going to talk about our software development, how we think about that, but we're also doing real breast cancer research or oncology research. We think about cancer health disparities and how we can use spatial biology to leverage that. And we also do technology implementation, development, and so on. All right, so why spatial biology and what is spatial biology? So first of all, why is it the hottest field in biomedicine? It was that in 2020 when it was called Spatial Method of the Year, but it's also still the fastest growing number of data sets. And that was like 2 years ago, but it was last year and it's also this year, and it keeps on growing and growing. When will it transform? That's also one of these questions. When will it transform research and clinical practice? And how can I enter? It's a question that they ask me, like, what should I do? Which technology should I use? It's not an easy question. This is actually not a text from me. This is a blog post from Yanis Vlachos, who is the spatial director at Beth Israel Deaconess, and he's doing a lot of really cool research. All right, so why spatial biology? I'll just gonna jump right into, there are many different reasons. I think the most obvious reasons, obviously, like it gives you spatial information, while the ones that the counter, let's say the counter technology, single cell doesn't really do that. It doesn't have the limitations that single cell RNA-seq does. I don't know who of you does single cell RNA-seq, but there's a lot of like dissociation bias and other things that you cannot really have. It gives you a large number of cells. It has a high number of sensitivity depending on which technology you use. And really everybody has their own like favorite way why you should go into spatial biology. My favorite one so far is the fact, and this is just that it can really be applied on virtually any tissue, including any clinical tissue. And so I think the translational value is so much bigger than some of the other technologies that I've previously played around with, including high chip, nascent transcription, ChIP-seq, cut and tag. I did it all. I'm just the most impressed by spatial transcriptomics or spatial omics in general. All right. So how does the field typically adopt a new spatial or a new technology, in this case, spatial omics? So we develop technology. Many different labs have developed all sort of really cool technologies. At some point, this is being translated in commercial platforms that are available to everyone, including all of you. They make us think. They make us biologists think about what can I do, what are the samples I have, what are the questions I can answer, and then we do all sort of cool data analysis. Think about how the spatial niches play a role, ligand receptor signaling, spatial patterns, everything you can think of. Today, I'm going to talk about something that is oftentimes forgotten and I think is really important. And it's the fact there is this in-between component, which I call software engineering, which I believe is essential if we really want to make this a sustainable environment and something that we can all benefit from in the most optimal way. All right, so what exactly do I mean? And this is a paper that was written by Anna Carpenter, the original developer of Cell Profiler, if you know what that is, and Shantana Singh. And it's bringing computation to biology by bridging the last mile. And she has some really interesting points. It's a paper in Nature Cell Biology, very short, but I all encourage you to read it. I'm just gonna read out loud two of her paraphrase two of her like sentences. So first of all, usable tools are oftentimes the last mile bridge between what a computer science or a bioinformatician makes possible and what biologists can really put to widespread use in their research. And everybody can develop tools, but putting them out there and letting people use them is a whole different field. And then something else, rarely are usable biological software bridges built owing to the lack of dedicated resources and rewards for this stage. So this work really requires software engineering expertise, which is expensive and underfunded by funding agency typical mechanisms because usability and maintenance work does not itself constitute new research. And I think it's very unfortunate, and I decided to go right against that and do this type of work, at least for some part of my lab, even though, and I agree, it's very hard to get funded, but it's just the impact that it has is so much bigger than anything else we're doing so far. But it's also really necessary. So there are many different technologies, spatial technologies, as some of you might know, and we really need technology-agnostic structures if you want to understand these and use these technologies maybe together or for different projects. This is just to give you an idea, like all the different technologies that have been developed over the couple of years, and there are really many of them, and I'm forgetting many of them as well. All different technologies, different resolutions, different technologies, different. Sometimes it's RNA, protein. It can be lipids. It can be a little bit of everything. As I showed before, the number of data sets and methods exponentially increase. So if we want to build these type of software infrastructure that can support spatial data analysis, it's now or never. If we're going to wait another decade, then it's going to be completely fragmented, as we did with some previous technologies. And perhaps most importantly, why do we need to work on software engineering approaches? Why spatial data engineering, spatial data structures? It's because the data is really complex. It's much more complex than any other type of data you previously worked with. And I'm going to show you why. And I think this one is a particularly really bad example. It's this idea where bulk is RNA-seq, single cell is kind of like the kind of like the milkshakes or whatever. A single cell, the individual ingredients, and then you have this pie. But in reality, spatial data is so much more complex. So you shouldn't be actually giving even like one of these examples, but I'm still going to do it anyway because I'm kind of stubborn. But I'm going to use a different approach. I'm going to say that RNA-seq is kind of like you look at very old bricks, and what can bricks tell you about something? It can actually tell you a lot. Our ideas about the Greek empires and the Roman empires, a lot of it is based on the old ruins of what you can teach us. But it's limited. So if you really want to understand like why is a building and what does it do, you really need to be able to look at these individual components, and it can be the glass, can be the bricks, can be anything that constitutes a house. That's a single cell, kind of like in a very simple way. But what is the actual house? You need to put it back together. And each of these individual components, when you put them together in different ways, you can have different types of houses, different types of functions. But that's only one small layer of what I would call spatial omics. In reality, these houses are only a part of the bigger structures you might be looking at. And you also need to put them together in bigger cities. And these cities might also be organized differently. And the roads might be organized differently. And how you can access these cities or not access these cities can be very different. That gives you pretty pictures, but doesn't really give you insights in spatial transcriptomics. So in order to do that, you also need to be able to provide additional annotations. And then you need to, on top of that, you need to be able to make a more abstract representation so you can actually distill the information you need and then use it in any further hypothesis testing. And that's exactly what we're trying to do with Giotto. We're trying to find a data structure, a software that encompasses all of this so that we can use it to the fullest extent. And are we making these data sets that will benefit from these type of like very rich type of data structures? And I think the answer is yes. And just to show you some of the more complex data sets that I found, and this is already outdated. This one is the one from 10x, where they combine on serial sections. They did their Xenium platform, that's their spatial subcellular transcriptomics technology. They did Visium, that's their spatial kind of like sequencing-based technology. They did IF data, all of it on serial sections. There's another one from a Belgian group. I'm from Belgium, I'm a little bit biased, where they did kind of like lipid type of analysis, including with Visium, including with H&E, morphology staining. There's a very beautiful data set that I've looked at a couple of times. It's recently actually published. This is their like their bioarchive paper where they combine on serial sections. They did merfish, and the other one they did electron microscopy. So think about how to incorporate morphology at a very high resolution with Merfish, and I think that they saw some really amazing things. And then just to give you an example of one of the things we do in the lab, as I said before, we are very interested in cancer, and specifically breast cancer. And what can we do with the limited amount of samples that we have? And these are these very tiny core needle biopsies that we get in the lab, and then they basically tell us, figure it out. If you want to do single-cell RNA-seq, and we made big single-cell RNA-seq data sets from these biopsies, you use a whole biopsy, that biopsy is gone. You get maybe 1,000, 2,000. If you're lucky, you get like 5,000, 6,000 cells, and that's it. Now, you can also take one of these biopsies, you can embed it, and you can serially section through the whole biopsy. You can easily have more than 100 sections. From a single section only, you can have about 40,000 cells in a single section from a triple-negative breast cancer biopsy. So we actually made a 3D, what I call a digital 3D biopsy, where we serially sectioned, and we have like 8 sections through the whole slide, and we have like in between, we have additional sections. And this is just an example where we created a Visium data set, but then we also have additional slides in between additional sections that can be used for, you can ask your pathologist, can you annotate the data? You can do IMC staining, you can do additional IF stainings and so on, and then start thinking about how we can integrate all the data to really try to understand like why is that cancer so aggressive, or what happened with that specific patient. All right, so the central question now, we're actually getting to the actual Giotto Suite tool itself. So how can we build a unified and standardized data structure to represent and combine all these different technologies and data sets in a very efficient manner, and make it so that everybody can do it, everybody can analyze it to its fullest extent? And that might be like, what exactly are you talking about? Hopefully I can show you in a few slides exactly what it does. So we developed Giotto Suite, and this is where you can find a little bit more information. We have a very rich website, including with ideas about how to contribute, and lots of different tutorials, example data sets, and so on. So it all started when, at the end of my postdoc, when I developed Giotto, which was a tool to analyze spatial data. It has a lot of new users every month, about 1,000. It was also the first spatial network, I think, at least on the bioRxiv, that was available. And back then it was very simple, and at some point I actually thought, like, I figured it all out, we don't need to do anything else. You generate the data, you have your spatial coordinates, you have your gene expression information, you put it in a simple data structure, and that was Giotto, and you do some kind of like data processing. We realized that it was limited to some of these problems that I discussed earlier, where you have multiple data modalities with different resolutions, with different inputs. So we're now changing this and making Giotto, we made it completely modular, so that all different components, like the visualization, the processing, the statistics, is all part of different components. Importantly, and it was because this is, we have different data representations that can basically report everything. And we also started thinking about different ways to store the data so that we can make it more scalable. All right, so I'm going to give you a few examples. I'm going to talk about how we have a different concept about how we represent the data, the spatial data. I'm going to show you some examples of what you can then do with multimodal data, what you can do with multi-scale data, how it can help you register multiple modalities, how we are interoperable with other very popular systems, how we can help you be interactive, so it's kind of like a human in the loop type of analysis, and some of our recent efforts in making it more flexible and scalable, because the spatial data is huge. The data is much bigger than any single-cell RNA-seq or combined single-cell RNA-seq data set that you've ever worked with. All right, so first, the data representation. So what we try to mimic in our data structure in Giotto, so kind of like an R package that represents data in a certain way, is we think about biology is kind of like multi-scale by default, but the technology that tries to profile these different layers of biology is also like multi-scale and with different types of resolutions. You can go all the way up from a big tissue down to the individual cells, individual organelles, and even individual transcripts, as some of the spatial subcellular data sets can give you. And there's different technologies that can profile these levels at different resolutions. These technologies also give rise to different representations, data representations. At the simplest level, you have images, but you can also have points, You can have intensity data if you have spatial proteomics data, for example, or spatial multiplexing data. And you have all sorts of shapes that you introduce yourself, or these can be cell segmentations, these can be whatever it is. And then we also have to think about how can we in the future then integrate all these different modalities so that you can easily connect them and start doing more like multimodal analysis. All right, so what we do is we completely separate the features, and that those are the data modalities, from the spatial units, and these spatial units can be your individual cells like you have with single cell, but in spatial you can actually think about spatial units as anything you're interested in. This can be your individual organelles, this can be your nuclei, this can be your cells, this can be your bigger neighborhoods, whatever you think of. So this is what I just discussed. You have the features, they can be anything, they can be spatial units, and then it's really up to you to think about how do I want to integrate these different features with the different spatial units. Do I want to do it once? Do I want to do it twice? Do I have multiple different technologies that I want to combine? Do I want to aggregate them at different levels? Do I want to combine them at different levels? Basically, everything is possible. All right, so this is kind of like the software type of approach. So now, can you please show me some examples of that? Would really be great. All right, so the first example is the idea about this kind of like multi-scale integration. So this is a data set from MERFISH where you, with very few efforts, can identify the individual nuclei. You can identify the cells. You can also identify niches. You can identify different domains with different types of spatial algorithms or tools. Now, how exactly are they all related to each other? So these are the type of questions that you all have at some point where you start analyzing that data, but it's actually quite hard to do very simple analysis and kind of like get to the data and compare the data as you want. So we make it very easy to just kind of like see like what is exactly the underlying relationship between these domains, the niches, the cells, and the nuclei. And then if you might have a question, we're looking now at MERFISH data, which is spatial subcellular. So you get like information about individual transcripts at 150 nanometers. So you can exactly see, are they located in the nucleus, in the cytoplasm, outside, wherever they are. So as another example, what you can then do as a next level is think about like how can we compare the cell versus the nuclei, and simply ask ourselves the question of which transcripts are enriched in the nuclei versus which ones are enriched in the cytoplasm. And for this specific example, is a breast cancer sample from MERFISH data. We see that in the nuclei, we have enrichment for transcripts associated with cell death. So maybe we're looking at a region that is kind of like apoptotic or necrotic. And on the other hand, the transcripts are enriched in this specific field of view, because you can do this with multiple field of views in a more informed way. There's enrichment for membrane-specific transcripts. So I think at least that kind of like makes sense in what you expect for this type of spatial data set. One other thing that I find interesting now that you have that data, so some people do single nuclei RNA-seq, others do single RNA-seq, and we basically have this idea that they are interchangeable in some way, that they give us like similar information, maybe one is a little bit sparser than the other, but that's it. So with spatial data and with Giotto, you can easily generate a kind of like a pseudo single nuclear RNA-seq data or pseudo single cell RNA-seq data, jointly cluster them, and then ask the question, how do these cells, if you cluster them in kind of like an unbiased way, which you would normally do, are the nuclei and the cells matching with each other? In other words, are the cell states that you get from a whole cell and the nuclei states, are they the same? And I think you can easily appreciate that it's true to some extent, but not for all of them. So when you're looking and you're clustering your nuclei versus when you're clustering your cell, this is like one of these examples where you can identify that's not always exactly a one-to-one comparison. And you might want to think about what exactly you're looking at. All right, then I'm going to give you another example, a couple of examples and this is now where we enter the whole idea about spatial multimodal registration. And so feel free to interrupt if you have any specific questions or want me to kind of like stop and kind of like guide you through everything. So we used the 10x data set because it was publicly available, and it has this very interesting idea where we have from adjacent breast cancer section, we have a xenium. This is a spatial subcellular data. We have IF data, and they have like, I think 2, and now they have like up to 6 different antibodies or proteins. And we have physium. So you can use any tool to register them, and we did it ourselves. We gave an example, and you can then provide that into Giotto, and you have kind of like a data set that is, or Giotto object, I should say, that is represented, where all these technologies are together. And now we're trying to make it as easy as possible to kind of like cross-compare, correlate, do everything you like to do with those different technologies and all the questions you might have. So one of the questions we had, and it's a really important question and something that hasn't been resolved yet, like what is the effect of the segmentation tool you use? So when you have these kind of like xenium data, Murphy's data, cosmics data, whatever subcellular data, you need to decide on the segmentation. So you need to kind of like have the cell boundaries in order to assign the transcripts to the individual cell, and then you say this is cell A, this is cell B. So we had this idea like, what if we compare the original cell segmentation provided by 10x, and we use another segmentation, and we use Baser, which uses the DAPI statement. On top of that, it actually uses the transcript itself. And simply ask yourself, what if we jointly cluster that? What are the differences we see between using one segmentation tool versus the other one? And this is what you see here in terms of like the cell type proportions. And I'm just highlighting the one that is the biggest change, and there are a couple of them, but we see a really big increase or decrease in the stromal and the cancer-associated fibroblast using one cell segmentation versus the other. And I think it just illustrates you that with Giotto, at least, you can actually use any type of cell segmentation, jointly cluster them, and kind of like figure out like what is important there, what am I missing, or also just trying to understand should I be merging them or not. But these cancer-associated fibroblasts, they play pretty important roles in breast cancer progression that was recently established. So understanding the limitations of your cell segmentation might also be very relevant in this specific case. We did something similar, and I'm kind of like going in a slightly different direction. This is IMC data, this imaging mass cytometry data. So this is spatial protein data, so it's intensity based, but as I showed before, we can also just incorporate it, put it into a Giotto object and analyze it as we did before. Now we're doing a similar but slightly different exercise as we did before. So we have our individual cells, they have individual segmentations, we cluster them, and then you see these individual cells, and they all have a specific cell type label A, B, C. Now, what we did is a very simple approach is where we said, what if we now, because you have complete control over these polygons, like all these objects are part of a Giotto object, we can shrink them, we can increase them, we can ask what is overlapping with other cells. What if we simply shrink all these cells by 25%? And you might actually say, why shrink them in the first place? Well, IMC data is actually known, it's a very naive approach, but IMC data is known to kind of like scatter and to cause cross-contamination from one of the cells to another, so that beta cell might become an alpha cell if you were looking at pancreas. So it might be a very naive approach to say, like, what if I kind of reduce contamination from the IMC, the laser, and the ablation, the scattering? What we see is that if we do this, and we redo the analysis, that we actually have up to 25% of the cells, they kind of like have a different clustering. They don't go to their main clustering anymore, indicating like how important it is to understanding the limitations of cell segmentation and using this IMC data. So this IMC data is being used in some of the biggest breast cancer data sets of the world, where they associate these neighborhoods with survival outcomes. So it's very relevant because like exactly what I'm showing you right now is if you look like where are these cells that are now have changed their cluster just by shrinking them a little bit, it's not too crazy. They're all over the place. So that means if you use now specific neighboring clustering algorithms, you try to associate it with disease outcomes, almost all your cluster algorithms your niche clustering algorithms might be slightly off or might be slightly different, and you might have completely different results if you use a slightly different segmentation approach. All right, so that's that part. And this is all just kind of like showing you what Giotto can do in a very easy way with a couple of steps. So you can all do it, and it's not very specific to one or two labs in the world. One other thing is, as I said, since we can collapse all these different data modalities with all these different spatial features, you can just generate your own data and compare any type of spatial modalities, including what we're doing here, compare between protein RNA, or maybe you want to compare Visium with Xenium. And that's something we did here. So this is simply protein versus RNA. Since they only had like two IF data set, one for CD20 and one for HER2, there's not a lot we can do. But just visually, we could kind of like confirm that there was some agreement We can also put a statistic to it. I think a slightly more interesting one, what we were very interested to see is xenium and visium. Do they provide the same information? Is it correlating with each other if you look at the same gene? So we did like gene by gene comparison between xenium and visium, and this is now for the gene FASN. You can just see by eye that it's actually pretty good. So that's one of the top contenders, like very high correlation values. It's just a Pearson R, a spatial Pearson R. You can see that it's very high. It does drop. We see a drop for all the different genes, for all the overlapping genes that they have between them. But then we're also wondering, like, why is it dropping? Is it dropping because like visium and xenium are just different? They're capturing these transcripts different. But what we just see if we look at the xenium expression levels and the visium expression levels, that they actually also all both drop. So it's very like you can easily understand that if it's very hard to capture them in both of them, you're going to look at a lot of noise, and so your correlation values are going to drop down. So overall, I think xenium and visium, at least in terms of like the genes you get out of it, is very nice and it's very beautiful, and it correlates quite nicely. One thing is a joint clustering, and this is now where I'm giving a kind of like going over this really quickly, but like you might have like RNA data, you might have protein data, and this is the visium site assist data where you can have information about up to 50 different proteins and your whole genome. And you might want to put it in a network. You use a weighted nearest network analysis to kind of like aggregate the two and combine the two. And that's what we do here. I don't want to really go into detail. The bigger idea is more about we can represent multiple different data modalities. You can aggregate them the way you want. You can use a weighted nearest neighbor analysis or any type of other like network integration analysis to look at multiple modalities and how these modalities define different cell types that you might find or the biology that you might find in your specific example. All right, so the interoperability, I mentioned this really quickly. I just think it's kind of important in the bigger space of software engineering. So on the left side, we have Giotto. We are at least interoperable, although we have some issues from time to time with three major software engineering approaches. One of them is the spatial experiment from the bioconductor research group or the bioconductor group. The other one is SCSAP, and then the last one is the N-data framework that is used by SquidBio. And we're now working a little bit on the new spatial data concept that the people from SquidBio and others are using to represent spatial data in the future. This is just an example how you can go from Giotto to N-data and then use any tool that is developed on N-data. For example, Bento is a tool developed in Python to look at subcellular organization or subcellular transcript organization within individual cells, which is something we didn't have in Giotto, and we just made that connection through their N-data framework. So you now have access to that specific tool, and you can think about how your transcripts are subcellularly localized and how that might change if you do, for example, a certain treatment or not. All right, and this is one of what I find one of the more exciting things, where what we try to do is, can we connect Giotto Suite with the existing spatial statistics world that has been around for like decades? Like R has been actually very strong for decades already, doing all sort of different spatial statistics works. Geospatial, you can call it. So what we did is our objects are actually, they are based on Terra, which is one of these frameworks, but we make them so that our vectors are point data. We can easily convert it to stars, to SF. These are some of these other big spatial statistics frameworks that exist in R. So you can move to that field or just bring in any of these technologies in Giotto and use their type of analysis that are available. And there's a lot available, which, and this is what I'm seeing here, is if I have seen further than others, it's by standing on the shoulders of giants. And these packages that have been around are really giants. And I think we should leverage it. We should use it. We should think about how we can use that for our spatial omics field. This is just to kind of give you an idea, like this is just for Terra alone, but you have all these other tools that we now have connected with Giotto and what is available for you to use in terms of spatial statistics, spatial data analysis. I'm just going to give you one example how we, in only like a week or two, use some of these data sets to do something with our Giotto object and to enhance some of our data sets. And so that is this specific example where, So we now have Giotto Suite, and this is an example of the mouse Visium brain data set, where one specific gene, it's called Panther1. You have this typical spot type of resolution where Visium gives you 55 micron spots, and then there's some empty space and you get another spot. So it's very like coarse resolution. It's not that great, although it can give you an idea about spatial data. But what we can do, as I said before, we can actually use the H&E data. The H&E data is with one of these images, we can use that. We can also identify segmentations using any type of segmentation tool, identify the nuclei. Now we can incorporate this segmentation tool into Giotto. So that's one step. So that's one way that we identify the individual nuclei within that specific data set rather than just using the spot resolution data. In addition, we use a process, what is called kriging. It's spatial interpolation. So it's a well-established process that is being used in the field with R, but actually in the weather and geology data to try to enhance, one of the things at least you can do is enhance the resolution of your images. And that's exactly what you can do with your Visium data, because your Visium data has like some spots where you have confident information, but there is lacking in between. And so if there's spatial variation in your genes, you can use that to say like, yeah, let's try to predict where that gene might be in between the regions and or even at a higher resolution than you had previously. So if you apply both of them together now, you can assign what we had previously. This is like where we assign gene expression to individual spots. You can now assign them to individual nuclei and have an idea about the actual structure and how these individual cells within your Visium data, what kind of genes they express. So that's one thing we did for one specific gene. You can also do that for all genes. And so when you do that for all genes, here on the left, you see what you normally would have had in terms of like resolution, where you have the individual spots, but on the right, you have the individual nuclei. We have enhanced our Visium data to individual nuclei. So we have H&E data with individual nuclei. We have all the genes, and now we ask ourselves, how do these nuclei cluster compared to the spots? And then it's kind of maybe hard to see, but it really nicely follows the structures, the underlying morphology structures of your mouse brain in this case, rather than having some kind of like very broad band of spots that are nearby your specific structures in the mouse brain. Yeah. So this is really the key part is each gene that you see here. So each gene has a spot, and a spot level has a certain expression level. We convert that gene into images at a higher resolution using the interpolation strategy. So now the input is not anymore the spot level expression data, but it's just this enhanced image data of that specific gene. So now we have, instead of having like, I don't know, we used the 500 genes, we have 500 images, enhanced super images that are interpolated. So you kind of like predict what is in between all these different spots, but also at a higher resolution. If you follow the spatial gradient or a spatial covariance structure, you can kind of predict what is an individual resolution of, or individual pixels of that that image that you're trying to predict. So that super enhanced gene image. Now you can overlay that with your with the polygon information that you get from the H&E. So you can identify these nuclei. And rather than assigning the transcripts to the spots, we try the image information to the individual nuclei. And that's how you get nuclei information. So the nuclei is basically a bunch of intensities. It receives a bunch of intensities from these images. And that's what we use as input. Yeah, maybe I should have provided a better example. So that's that part. What we are not doing, and what we actually hope to do in the future as well, is also incorporate the morphology information itself, so that you, in addition to having the super enhanced gene information, you can also like use the actual morphology information, and gives you a little bit more a confidence that what you're looking at is accurate. All right. So one thing I think the whole spatial field is probably going is having a more interactive type of data analysis. spatial data, much more than single cell, really benefits from your own eyes. You do some type of analysis, you use an approach, you look at it, and then you have an idea. You see a structure, you see something. You want to use your own eyes and identify, this is an interesting region. I want to compare this region with that region. And that's something we have a bunch of like shiny apps that you can quickly launch. You address regions, or you highlight regions that you're interested in. And then that information is directly available into your R console as a reactive object. So what that means is you don't have to go outside and store your object and go back and read it in and read it out and then write it out. You can, when you're working in your R Studio or whatever you like to use, you can launch it, do something, and that's available. And you can do, for example, differential expression analysis, or you can do anything else you would like to do. Similarly, if you have a 3D data set, so that's one of the data sets that we're generating, but others are doing that as well. We have in silico slicing tools, so you can slice your data in any way you want, which is just a very simple way of generating some in silico data and seeing like what would have happened if I look at it this way or that way. Would my observations, would they have changed or not, or would they have stayed the same? All right, and then one final thing is looking at what I said before is flexibility and scalability. And I just have a couple of slides. So the data is really huge. Looking at, for example, spatial genomics data, the nanostring cosmics data, the SCSAP data, we have implemented like some state-of-the-art methods from the bioconductor community, including the delayed array, which allows you to do delayed operations on a matrix, for example, and implemented parallelization. But even then, some of these operations are almost impossible. So one thing we started doing is we implement our own spatial chunking approach. You can apply some of these methods on chunks and then aggregate them all together. Another thing we did is make Giotto available on the cloud so that you can leverage some of these cloud processing. But I want to give you another example at the end, but just to kind of like show you what our flexible, and this is again going back to the spatial units, where these spatial units can be anything you want. So if we look at stereo-seq data, but also actually Visium data and the new Visium HD that is coming out, it's up to you, the user, to think about what is the resolution you want to look at. How do you want to aggregate the data? So one easy thing you can do if you have a very simple or a very straightforward way of aggregating data, and this is exactly what Giotto provides, you can aggregate the data first at larger scale, do some preliminary analysis, look at what is available, and then zoom in. And that's what we do here. We first did it at a larger scale. This is the stereo-seq mouse embryo data. It's massive. It's very hard to do. But this type of analysis can be done on your own local machine. If you use first like a higher organization strategy, you identify the region of interest. Now, in the same Giotto object, you say like, hey, let's use a higher resolution, like smaller tiles. What can I find? And then all of a sudden you can find more structures because you're using a smaller strategy. You can visualize the individual transcripts. Another thing that Giotto, and I'm just kind of like giving a sales pitch for Giotto, right, is that a lot of these tools, you can use data like this to generate pseudo-Visium data. And why is that important? Because I think if you use Visium data, you probably are also using some kind of like deconvolution tool, because you have single-cell RNA-seq data, you want to deconvolve each spot, but how do you know which deconvolution tool works best? You can use this type of data just to kind of like assess this, because now that data gives you the ground truth. You can make a pseudo-Visium data set from that ground truth data set, and then just try whatever benchmarking or deconvolution data you want, and you will see the actual results of that deconvolution algorithm, and you can directly compare it with the original ground truth data that you got from this type of technology. So that's at least one way you can assess yourself for your specific data set, for your specific single-cell RNA-seq technology. What is the best approach that I can take to deconvolve my Visium data? All right, and then, but if you really want to make dealing with this data set, you want to make it scalable, and you want to make it so that it's democratic, so everybody can do it, we need to think really out of the box and think much bigger. And the data can be big, right? The data can be big. So a single data set that I showed you, single xenium data, Murphy's data, cosmics data, whatever data is massive and is already a problem. Now think about what many of you are trying to do, including my lab. We try to make massive data sets, maybe with 10 samples, maybe with 100 samples, then it becomes almost unbearable. So this is why we also started thinking about a bigger idea, which now is called the DBverse, where you analyze your data as if you're working on your R package, but in fact, it's actually backed by a database that is scalable, and it's actually scalable on your local machine. So now you can analyze data that are up to billions of reads. You can do it on your local machine, so you don't necessarily need to have that like very fancy infrastructure, although it might still help. And so we're working on some like lower level representation, actually lower level packages that can represent a matrix. That's important because the matrix can be big, but also that presents the different spatial geometries that I discussed, the spatial sequence information if you have spatial sequence information. And by using that and leveraging a database, now Giotto Suite, and this is not yet available, I'm sorry, but that in the future, hopefully we can scale up to even like analyzing the biggest data sets that you can think of. This is just giving you a quick sneak peek as if you can, and this is also, I think, one of these StereoSeq data sets, and we have a small region of interest all the way up to a whole embryo. And some of these spatial operations are matrix operations, including like identifying transcripts to individual cells, looking for joint overlaps between cells, for example, or whatever, and doing all sorts of operations DB matrix. That you can actually see that for many of these other tools, state-of-the-art tools, that would even consume huge memory, it would be out of memory, it will never work. But with a matrix or a database-backed approach, you can actually do that in a matter of oftentimes just seconds and maybe a couple of minutes if it's really huge. So I think that's really exciting and something that we're looking forward to. So I have one or two more slides after this, but this is just making sure that I can thank everyone. So this is really a major effort between my lab and Giotto's lab at U1 lab and at Mount Sinai, where Jocelyn and George in my lab are like the ones driving the whole field, are driving the development of Giotto Suite. But they also got a lot of work, or they got a lot of help from others in my lab and in Giotto's lab, because there's a lot of people that are using Giotto, and a lot of times they have a bunch of questions, and there's like problems here and problems there, and they're emailing us, they're putting things on GitHub. So it's quite a big effort. So we try to kind of like we have a lot of funding that helps us, and we have bits and pieces everywhere so that at least we can support some of the people working on this full time. So we have a preprint now for Giotto Suite. It's on bioRxiv. But last but not least, I just want to basically say like this is where you can find more information. If there is a bug, then please let us know, and we'll try to fix it as soon as possible. I am actually trying to set up a more like open source governance structure for Giotto, so that's not just my lab and Giotto's lab that is kind of like pulling the strings and telling what to do, but it's more driven by the open source community. And one thing I'm actually interested in, this goes back to, and I don't know if some of you are working on something similar, it goes back to the interoperability, where I'm hoping to have some kind of like, can we define a spatial data framework, like a metadata framework, where it's not just like you have to use this or that data structure, but more about this is exactly what we expect. This is the spatial metadata that you need, even if you have multimodal data. And if you know that kind of metadata, then all developers, including us, people from Saras, people from spatial data, people from bioconductor, can adhere to that, and it will be very from one technology or one data structure to the other. And that's it. I'm just going to go back to this one. Yeah, if you have any other questions, let me know. Yes.
Couple of questions. First one, going back to your comparison of segmentation. One of them is right and one of them is wrong, or that segmentations are all really good.
I hate picking one out, so I'm not going to do it. It probably depends on the data set you're using, the additional information you're using. The one that people seem to like is self-post, but also a lot of the others are really good. But they all seem to miss some key cells. And if you actually look at your raw data, a lot of times you see like very clear signal because you see like accumulation of transcripts. But if there is no DAPI or no nuclear stain, it's just simply most of them are not going to pick it up. So I think a combination between some of the more popular imaging-based segmentation strategies with segmentation or imaging-free segmentation strategies that can work directly on top of the transcript information, and a combination thereof is probably the way to go in the future.
The question is about the try to establish cross-disciplinary collaboration?
Yes and no. Actually, I talked to someone in geology, I think, and he was actually interested to talk with us. And it's just so spread so thin right now that I haven't reached out to that person again. So it's probably my fault. But it's something I'm thinking about, and I've mentioned this to a couple of other people. But in the next couple of days, I'm going to attend a mini labs from the NIH to think about cancer health disparities. And this is something that I would like to bring up, like how can we bring more expertise from these sciences that have already been doing this for years, and can we bring them into spatial omics field and help us think about what should we do, and let's not make the same mistakes as they made for like 20 years ago. For dbVerse, is this similar to the BP cells framework that now is supported by Surat for single cell RNA-seq and ATAC-seq data? Right. So the question is if the dbVerse and the db matrix, I think specifically, is that similar to the BP cells framework that is supported by Surat? So I know that BP cells is trying to do something similar where they also have another way of representing their big matrix data so you can analyze it. I hope there's no one from Surat on this call. So we tried to use it. We tried to compare with it. The results were pretty bad for BP cells to the extent that I'm afraid that we implemented it wrong. And I asked my student, can you repeat it again? Was the same problem. So I'm not sure if we did something wrong. So I don't want to share this until we're absolutely sure that we did this right. But yeah, it's the same idea. Can we do something more scalable? Except we cannot really get BP cells to work. Again, it might be our fault. I don't want to blame them. So yeah. So you mean this? Are you talking about the shrinking of the polygons, basically the cell boundaries? We don't actually lose the cells. The neighborhood change. Like if you in a very simple way, right? If you think about the neighborhood, which is simple, a cell type composition of multiple cells around them, whatever, that definitely changes because we do see that some of these cells change their main cluster that they were associated with. So that definitely changed. And unfortunately, these cells are also all over the place. They're not in one specific spatial area, because then you could say like, yeah, that's for whatever reason, these cells need more information or less information. That just also indicates that almost every single niche, if a niche is comprised about like 5, 10, whatever cells, would have changed a little bit. And I think that the more important thing is that now we are trying, and not necessarily us, but others, we are trying to associate these niches with survival outcomes. Think about like where they say like you need a CD8 cell with a DC cell or something. But if it's that sensitive to changing the polygon data, like the cell size, then we need to be very careful at least to have like very strong conclusions attached to it. Does that make sense? Or is it, yeah.
What is the problem, the multi-scale problem? When you have multiple samples across patients or multiple modalities per sample across patients, what's your preferred strategy for data harmonization and data?
For different sections, like different sections with different, you mean which tool that we prefer to use?
How do you like to harmonize? Like, there was a lot of effort around batch harmonization from single cell problems, like finding right landmarks or whatever it is.
Yeah.
How do you typically try to integrate slices of different modalities?
Yeah, I think we are working on our own strategy, and again, more making it as easy as possible for someone using, for example, Giotto, so that you can just do it yourself. But the one that we have used and that we're quite happy about, it took us some time to get to, is the ST Align, I think from Gene Fenn's group, from Johns Hopkins, right? They have some pretty cool tools that allow us to do this. They also have like multiple different, because there are many different methods of registering. Some of them are a little bit more freedom than others. But SD-Align has worked pretty well for us for this specific data set. But this is also just an emerging field, I think. Like, how exactly do you do this? I wonder if there's also like other fields that have done this previously. They probably did. Although I do think that how with the whole spatial omics field, it's slightly different. There's a lot more homogeneity between, like, finding landmarks, for example, is a little bit more challenging. And there are a few tools that are out there, coming out there, that does this in a more unsupervised way, where you don't actually have to do it yourself, or at least with some semi-supervised way.
If you have data from different platforms, like imaging, calibration of the acquired data, sense for what's a good way of buying data.
No, this is an excellent question. I think this is something that we've been thinking about ourselves. So right now, our framework allows you to merge the data, and that's the first step, and allows you to easily like aggregate them, extract them, do everything you would like to do, and I showed you some of it. But how exactly, and how to what to watch out for when you do it, I think that's another field that we need to work on as a group.
You asked, how is Giotto different from other tools?
How Giotto? Yeah, good question. I think other tools do probably similar thing. I do think that from a high point of view, I think we're very comprehensive. I do think, and now I need to watch out again. I do think Giotto is probably the only one that presents both the data analysis and a specific data structure, where it's all kind of integrated. At the same time, as I showed you before, and didn't really stress this out, Giotto is kind of like modular now, so you can take out whatever you want if you're more specific in one module versus the other, or if you want to use the whole package. So I think that's kind of like where we differ. And then, what specific types of analysis can the spatial chunking approach facilitate? Good question. I think right now we used it mostly, I think, for like spatial patterning, I think, like spatial pattern analysis, and it's something that we still haven't fully implemented for everything. So that's still on our table and something that is on our to-do list. All right.
I want to add that as an R user, I'm especially influenced by Giotto. A lot of the tools for spatial transcriptomics are in the Python space. SciPy, SoPy, all those kind of things. Thank you for putting the envelope on R.
Yeah, we actually we are, and I should catch up again. We are talking a little bit with the people from SciPy, and so spatial data, because it's all related. It's now part of the bigger SE-verse. In terms of like size of their group and my group, is day and night. So we have to be a little bit more strategic about where we put our efforts. So yeah, but maybe in the future we can. I wouldn't say catch up, because I think right now we're more on par, but they're doing some really amazing work.
Thank you, Ruben.
All right, thank you.